Now, I'm going to show you guys how the colon actually looks like the slide in your fingerland. Sorry. So this is how your colon slide looks like. So I'm going to zoom in. You guys should always know, for special histology, you guys always start at the bottom and go to the top. Start by looking at the mucosa. Try to identify structures that you know will help you differentiate like your slide from other slides, or to try and figure out what system you're in. By zooming in, the first thing that I see is I know that I'm looking at simple columnar epithelium with goblet cells, and I'm looking at a lot of crypts. I already know that I'm somewhere in the intestine. As I'm zooming in, I'm seeing that these crypts do not have any Paneth cells. I will know 100% that I am in the colon. And as I zoom out, one thing that I notice is this thick muscle layer that are completely surrounding here. Sorry. Your thick muscle layer, which they will ask you theoretically, if you get the slide, they're going to ask you, okay, like what is this? You guys are going to say this is smooth muscle, and they're going to ask you to be a bit more detailed, and then you guys could talk about how in the tunica muscularis externa you have your inner and outer layer, and how the outer layer is the longitudinal layer that's forming the tinea coli. uh okay, and this is it for me. Any questions? Okay.
So, all right, guys, we'll move on to the appendix. So these are the two slides you can get or in your slide box present. In my opinion, it's really easy to identify. You can see in this tunica mucosa layer, there's a lot of lymphatic infiltration, and you can see it has a regular lumen. So moving on, as I have said, that there's four layers. We have tunica mucosa, tunica submucosa, tunica muscularis externa, and tunica serosa. Now, when we talk about the tunica mucosa, it is made up of simple columnar epithelium with goblet cells. And as you can see at the tunica submucosa, there's infiltration of the lymphatic tissue as well. So the tunica submucosa, it is made up of lymphatic tissue, connective tissue, vessels, as well as nerves. When we talk about the other components of the tunica mucosa, which is the lamina muscularis mucosae, this is absent. This is not present because it is destroyed by the lymphatic tissue. And as you can see, the tunica muscularis externa, it is really thin, but it is still made up of two layers of muscles, inner circular, outer longitudinal. And then you just have the tunica serosa, which is the outer covering. Now, there is a chance you might see crypts of lubricant in the slide, but most of the time they're absent as they're destroyed by the lymphatic tissue. This is in fingerland. You can see one, or you can see one, two, three crypts of lubricant here. So mainly the differential diagnosis would be a small diameter, narrow lumen, as well as abundant lymphatic tissue with lymphatic follicles in the entire lamina propria mucosa. Now, when we talk about the 8 changes of appendix, here you can see these pictures are in your lecture as well. You can see the difference between appendix of a 10-year-old and a 36-year-old person. As you can see, the 10-year-old appendix, it has prominent lymphatic prominent lymphatic follicles, and this is due to the fact that they will have a strong immune function to protect them, and it has a thick mucosal layer, as you can see here. When we look at the 36-year-old, they have reduced lymphatic tissue. This is due to the fact that they have mucosal atrophy, which means that the mucus goes through degeneration, and it has an irregular shaped lumen, as well as the fact that there's more folds inside the lumen compared to here. Now, this is due to the fact that as the appendix ages, it goes through fibrosis. What this means is that this makes the tissue in the tunica submucosa appear more denser and less cellular. So if you zoom in into these here, the tunica mucosa of the person with 10-year-old appendix would be more cellular. However, when we look at the tunica mucosa of the 36-year-old appendix, it would be more denser. So
Any questions, guys? Okay, so we're going to the anal canal. The anal canal is something that usually like students do not struggle with, guys, just for the simple fact that you are not going to see any other slide that looks like this and has these characteristics. So first, I just want to bring your attention to here. This is an image that is found in your lectures. I think it's a very good image so you guys to be able to understand what is happening in your anal canal slide. As you guys know, your anal canal is what's going to pretty much mark the ending of your like GIT with like the outermost part of your body. And as you guys can see, you have a lot of structures that are like surrounding, which I'm sure you guys might know. We have our internal hemorrhoidal plexuses, we have our anal sphincters, then we also have our external anal sphincter, we have our rectum, our anal canal, and then our anal skin. So for your anal canal slide, this is a slide that try to imagine then that they cut like a section like this. They cut this section. And so you can see, obviously it depends on the slide, but it is very possible for you guys to see like the external, like muscle of the external sphincter, the internal sphincter, to see the hemorrhoidal plexuses, to see a little bit of the rectum or the epithelium that signifies that it's in the rectum. You're going to see the epithelium that's in the anal, well, I will show you guys in a second what I mean, but you're going to see three different epitheliums to signify these three different zones. So the anal canal is composed, just repeating myself, is composed of three zones. You have the columnar zone, the intermediate zone, and the skin zone. Columnar, intermediate, and skin zone. So the columnar zone, you guys, is going to have crypts of lubricant. It's signifying the part of the anal canal that is still like containing the rectum or like the same structures of an epithelium as the intestine. You're going to see simple columnar absorptive epithelium with numerous goblet cells. This is going to end abruptly. There's going to be an abrupt change, and it's going to be replaced by stratified squamous non-keratinized epithelium, which is going to signify that you are now in the intermediate zone. In the intermediate, sorry, in the intermediate zone, you're going to see stratified squamous non-keratinized epithelium. You can see some anal mucus glands, and if like this is a trick, if in the slide, if you get the slide and the professor points to a bunch of structures that you guys know are vessels and you guys know are veins, and he asks what is this, he's most likely pointing to the internal plexus, which will then continue like, so the internal plexus, you will see it in the columnar zone, and if you're in the intermediate zone, it's a continuation of the internal now going into the external. And you will also see the inner circular layer, which will become thicker, of the internal anal sphincter, which will then turn into the outer, like external anal sphincter. If you're then going to see the skin, in the area of the skin, which you will see the epidermis and the dermis. As you guys know, stratified squamous thinly keratinized epithelium. You're going to see a lot of your skin appendages like hair follicles, sebaceous, sweat glands. You will see continuation of the external hemorrhoidal veins, and you're going to see like some forms, some layers of the external anal sphincter. This is not mandatory. Like I remember in my year, it really depended on the slide that the student gets. Some students had slides where they could see the sphincters, some students didn't. Um, but again, columnar, intermediate, and the skin part. The next slide is really just an example. I didn't really even want to include this pictures for you because this is not how the slides are going to look for you in Smart Fingerland. But I just wanted you guys to perhaps this might be like a better visual for someone that the Smart Zoom might not be. So this is like the columnar zone. As you guys can see here, simple columnar absorptive epithelium. You can see here some crypts. And as you guys see here in the intermediate zone, you have a much different epithelium. And you can see that the change is very abrupt. It's a very abrupt change going from the simple columnar to the stratified squamous. So again, this change here where this arrow is pointing is this, guys. It's the change from rectum to anal canal. This is what you guys are seeing. And this is just an example of what would look like the internal anal sphincter, which you guys are going to see in the columnar zone and in the intermediate zone. And then the external, like you could see under the skin zone. The anal valve is something that also really depends on the slide. I personally do not remember the professors even asking about it, but perhaps it's something that you guys could keep in mind. But again, this image is another example. Columnar zone, obviously you can't see the epithelium too well here, but I'm hoping that you guys can see the different changes. The anal canal slide is the slide that has 3 different zones. Columnar zone, intermediate zone, which you can kind of see that this is definitely stratified epithelium. And then you have the skin zone, which you can see from here that it is the skin zone because of all the appendages. And you can see some like thinly keratin on the surface. And now I'm just going to show you guys the actual slide. So I'm going to start here, the outermost part. This is clearly the columnar zone, guys. I just need to zoom in, and it's no different than what we have been seeing before. Look at the simple columnar epithelium, look at the goblet cells, look at the crypt of Librekin. This is the columnar zone, again, columnar zone of your anal canal, and then boom, there's a sudden change, literally a sudden change from columnar to stratified squamous, non-keratinized. Now we know that we are in the intermediate zone, and then we go from the intermediate zone to, look at this, all thinly keratinized epithelium, which tells us that we are now in the outermost part, which is the skin part of the anus. So this slide here is, I remember, a slide that we had available for us, and these vessels here could be considered the plexuses. However, there is another slide for you guys, which is a new slide. Again, you guys can 100% notice the difference of these epithelia. And come on, look at this. And this is not something that you guys are going to notice in any other slide. You're not going to see such abrupt change. You look at this, you see all these different epithelium, and you know that you are in the anal canal. I think that's it for me.
Okay, so I'll be explaining you guys about the liver. Yeah, so the liver is covered by something called as the Glissen's capsule, which is made up of a mesothelium, and it covers the entire liver, and it also sends in septa into the liver, which divides it into lobules. But on the slide of the human liver, you cannot really see the lobules. You can only see it in the slide of the pig, which I will be showing to you at the end of this. There are 3 important vessels which help in the blood circulation in the liver. That's the portal vein, hepatic artery, and the hepatic vein. The portal vein brings in all the products which are rich in all the products of the digestion. So it brings it from the intestines to the liver. And the hepatic artery mostly supplies the liver with the oxygen, and the hepatic vein just deals with the venous drainage of the liver. So there is something called as a classic liver lobule, which is classified as a functional unit of the liver. Okay, so this hexagonal shaped structure, which you can see here, that is the functional lobule of the liver, and it has a central vein, and it is surrounded by the white space which you see here. That is the periportal space. So it is surrounded by this. Next, we have something called as the portal lobule. So that is the functional lobule of the liver. Okay, make sure that you stress on the word lobule because this is different from the functional unit of the liver. That is the liver acinus. But right now, I'm talking about the functional lobule of the liver, which is called as the portal lobule. So the periportal spaces, they contain something called as the portal triad, which includes 3 vessels. So there is the interlobular arteries, the interlobular veins, and the bile duct. It is in the center here, as you can see. So this is your portal triad, and like it gets the secretions out from 3 hepatic lobules, right? So as you can see here, there's 3 of them, and the things from these 3 is draining into this one lobule over here. One key difference to note over here is that lymphatic vessels originate from here and then they spread out into the liver outwards. But all the other vessels and bile and everything is coming from the classical lobules into the portal triad. Next, we have the liver acinus, which is a functional unit of the liver. Okay, it is found inside the hepatocytes. You cannot see it. It is just a theoretical thing which you need to know. It has got three zones. The zone one, it receives the largest amount of oxygen because it's present the first. Yeah, it's present right beside the portal triad, so it's going to receive most amount of oxygen and the nutrients and everything. So the hepatocytes, like in this area, they synthesize the glucose, the glycogen, and the plasma proteins. But since like the zone is the first, so it also comes in contact with toxic substances the very first, so it could get damaged. Next, we have the zone 2, but it is just like an intermediate zone between zone 1 and zone 3. It does not really have any specific function to it. And zone 3, that is like near to the central vein. So the hepatocytes over here, they are low in concentration of oxygen. So here they are mostly in charge of detoxification. And over here, they're also susceptible to damages which are caused due to low concentration of oxygen. For example, hypoxia could be caused. Next, we have something called as a liver plate. So in this diagram, you can see like there's a bunch of hepatocytes which are connecting, and it's this structure basically when hepatocytes connect and they form like a rod kind of a thing that is called as a liver plate. So the hepatocytes are surrounded by sinusoids. You can see this, for example, over here. So liver sinusoids, they are basically just vessels which carry blood into the liver. Two adjacent hepatocytes, for example, if we take this hepatocyte and this hepatocyte, so between them there is this tiny space which is called as a bile canalicus. It's not like a proper vessel. It does not have a lumen or anything. It's just a space between two adjacent hepatocytes. So the main structures in a liver plate include bile canaliculi, interlobular bile duct, and the Herring's canals. Next, we have the supporting vessels. This includes the central vein, which is over here, and the liver sinusoids, like I said. One important thing about the liver plate is something called as the hepatobiliary barrier. So it is between the hepatocytes and the bile ducts. It is made up of the gap junction. And the reason why this barrier is so important is because it prevents the toxic bile from entering into the bloodstream. Because if it does, then it can cause a bunch of problems. For example, it could cause jaundice. Next, we have the liver sinusoids in a bit more detail over here. So there are two types of cells in the liver sinusoids. Okay, there's the endothelial cells and the Kupffer cells. Um Endothelial cells are mainly in charge of phagocytosis, and they also have like fenestrations, which are like tiny holes. These fenestrations only allow the blood plasma to come inside and not blood cells. Okay, only blood plasma comes and it supplies the hepatocytes with all the nutrients and it takes away products from the hepatocytes away back into the bloodstream. Next, the Kupffer cells, they are the macrophages and they are of mesodermal origin, which you don't really need to know in details right now. You can do that later on when you do embryology. But yeah, they have these long processes, and Kupffer cells are not connected to endothelial cells in any way. They're like a whole different unit on its own. Their main function is phagocytosis, and it transports a substance called as bilirubin to the hepatocytes. Varun will be explaining you guys what bilirubin is in the upcoming slides. And the Kupffer cells also help in protein synthesis. So the space of Dissay, okay? So when you look at the slide of the liver, which I will be showing to you guys soon, you will see a bunch of these white spaces between the hepatocytes, and that is the space of Dissay. The main function of it is to separate the sinusoids from the hepatocytes, and it has a bunch of microvilli which increase the surface area for the hepatocytes to absorb the substances easily. It also has these special cells which are called as hepatic stellate cells, and it could also have some connective tissue fibers. So the hepatic stellate cells, they are also called as ito cells, and their main function is storing of fats. So when you look at here, like for example, this is like a fat droplet, and it stores fats, and it also stores vitamin A. It also produces a bunch of things, which include the reticular fibers, the collagen fibers, and a few substances which help in the hepatocyte regeneration. Uh, Sometimes what can happen is that it can produce a lot of collagen fibers, which will convert these hepatic stellate cells into something called as the myofibroblasts, which will then turn into liver fibrosis, which is a serious condition. So this is just an overview, but I think it would be better if I explain it to you guys on a proper slide. Do you guys have any questions till now? Okay, so this is the slide of the liver. So on the top here, you can see the mesothelium. So this is the Glissen's capsule, which is made up of dense connective tissue. And then all of these structures, which you can see here, okay, like let me zoom in. All of these, they are hepatocytes. Like for example, this is one hepatocyte, this is the other. So the entire slide, you can only see hepatocytes. And this space here, this white space, which I was talking about earlier, this or this for example, that is the space of the sea. And over here you can see there's like a few bunch of structures which you can see in the space of the sea. They do not look like hepatocyte nuclei or whatever, but you can clearly see that there is something present in the space. So these are going to be your liver sinusoids with endothelial cells and Kupffer cells. So you can see that well over here. And another important thing which you need to know in the slide is central vein. Okay, so this is the central vein. Like I said earlier, in the slide of humans, you cannot really see the lobules properly, so you will not know if you are in the center of the lobule or like where you are. But this is one central vein over here, and you can see a few more others, but you cannot tell that they're in the center of the lobule. This is a periportal space, this entire structure over here. And like I said, it has three things, right? It has the interlobular vein, artery, and the bulk and alkali. So this is the interlobular vein. Usually in most of these periportal spaces, the interlobular vein is going to be your largest structure. And you can clearly tell that this is a vein because its lumen is like really big, wider, but it's like really thin. But over here, you can see that this is an artery because you can clearly see the tunica media is like really, really thick. This is the interlobular bile duct. The reason why you can tell that this is an interlobular bile duct was by looking at its epithelium. So here you can see that the nuclei are really like circular, and it's not, this is not the case with for example with the artery or even the vein. You can only see these distinct nuclei in the interlobular bile duct, and that is because it is lined by simple cuboidal or simple columnar epithelium, whereas these two are not. So this is how you can distinguish between interlobular bile duct or and an artery. So this is basically it, the slide of the liver. This slide is, as you can see, like there's nothing much to explain in it. So you really have to know your theory behind all of this, know all the details and all the things which I've been talking about till now. You need to know them because when the professors see the slide, they will maximum ask you about like the periportal space in the slide, but they will ask you a lot of theory about all the other cells which I've been talking about, the spaces and everything else. So you really need to know that well. And this is a liver of the pig. This is much better to see here because like you can clearly see that the septa are coming and they're dividing the liver into multiple lobules. You will be able to see the central vein properly here because it's supposed to be in the center of the lobule, right? So this is one lobule, and in the middle you can see a central vein. Rest all the information pretty much remains the same. You have the periportal space here with the portal triad and everything. This everything else remains the same. And yeah, that's it for my part.
All right, guys, moving on to some more theory about hepatocytes. So first, let's talk about some basic functions of hepatocytes. It's involved in protein synthesis, they're involved in metabolism and detoxification, and they're involved in hepatocyte regeneration as well. So when we talk about the protein synthesis, it takes place in the rough endoplasmic reticulum, and it is involved in production of plasma proteins. These includes albumins, globulins, and other clotting factors. And it is important to remember that when these proteins are produced, they're not released in the form of granules, they're released directly into the bloodstream by the process of exocytosis. And when we talk about the metabolism and detoxification, that takes place in the smooth endoplasmic reticulum. This includes the glucose metabolism, amino acid metabolism, lipid metabolism, detoxification, and vitamin metabolism. So when we talk about the glucose metabolism, it is involved in the glycogen synthesis as well as breakdown. For amino acid metabolism, it removes the amino group of the amino acids. As we know, amino acids have one amino group and one carboxyl group. So it removes the amino group of the amino acids and results in the production of urea. And it is involved in the formation of bile acids, cholesterol, as well as triacylglycerols. And it is also involved in the storage of fat soluble vitamins. These include D3, E, K, A. It is really important for you to remember just these vitamins, nothing else. Just remember it is involved in the storing of these fat soluble vitamins. And when we talk about the hepatocyte regeneration, so in humans, it is in limited capacity. It only takes place due to the presence of some hepatocytes which are already present. Otherwise, it's not possible. And it is possible in animals, but this has only been tested experimentally. And this basically takes place by the differentiation of the hematopoietic bone marrow stem cells. So when we talk about the gluconeogenesis and glycogen metabolism in detail, as I said, it occurs in the smooth endoplasmic reticulum membrane, and it converts lipids and amino acids into glucose. I'm sure you guys know glycogen is the storage form of glucose, and glucose is whenever we need the energy, the glycogen is converted into glucose and supplies the energy. When we talk about the protein production, as I said, takes place in the membranes of rough endoplasmic reticulum and the Golgi apparatus. And the deamination of amino acids, as I said, amino group is removed, produces urea. This enters the blood, and then it is excreted to the kidney, and from the kidney, it moves outside our body. Now, when we talk about the exocrine function of the hepatocyte, moving on from the endocrine, it is involved in the production of bile. So first, let's talk about the components of the bile. This includes water, mainly, as you can see, it's 97% water. Then you have primary bile acids, bile pigments, cholesterol, and phospholipids, and electrolytes. So when we talk about the primary bile acids, they are produced in the smooth endoplasmic reticulum, and these includes the cholic and chenodeoxycholic acids. And these acids conjugate with the glycine and taurine, resulting in the formation of bile salts. When we talk about the bile pigments, these include bilirubin and biliverdin. I'll explain what they are in the next slides. And when we talk about the cholesterol and phospholipids, they are essentially involved in just the emulsification of fats. Some electrolytes which are present, these include sodium, potassium, calcium, magnesium. They are mainly involved in maintaining the pH of the bile. When we talk about the bile metabolism and recirculation, so first of all, the conjugated primary bile acids, which are conjugated by glycine and taurine, they are broken down by the bacteria present inside our gut, and they're converted into secondary bile acids. And when we talk about the further process, 90 to 95% of the bile is reabsorbed into the ileum, and it returns back to the liver via the enterohepatic recirculation. And only 5 to 10% of it is released into the feces. So when we talk about the bilirubin formation and processing, so guys, please, this is a really important process to remember, and I tried my best to simplify it. So first of all, bilirubin, it is derived from the hemoglobin breakdown. So first, the red blood cells, they will break down, and this will be recognized by the macrophages, as Ria had mentioned, the Kupffer cells. So hemoglobin, it is taken up by the broken red blood cells. They release hemoglobin, which is taken up by the Kupffer cells. Now this heme, it is converted into biliverdin, which is the yellow pigment, and then this is converted into bilirubin. Now, bilirubin itself cannot travel across our blood because it is insoluble and it is toxic in excess. So inside the blood, it forms a complex with albumin, forming the bilirubin albumin complex, and then it is taken up by the hepatocytes. Once it's inside the hepatocytes, it enters the smooth endoplasmic reticulum. An enzyme called glucuronide transferase, it converts this bilirubin into bilirubin glucuronide. This is water soluble. And now this can be excreted into the bile canaliculi, and then it enters the bile, and then it reaches the duodenum. So as you can see, this is a simple flow chart explaining what I just explained. So red blood cells broken down release heme, and this is unconjugated bilirubin. Once it enters the bloodstream, it reacts with albumin to form bilirubin albumin complex. And once it enters the liver, it turns from unconjugated to conjugated bilirubin. And then after from liver it enters the gut, where 80% of it is excreted into the feces, 20% is excreted into the urine, and 18% is taken up back by the process of enterohepatic recirculation. And then when we talk about the different biliary tracts, so they're divided into three parts. These include the intrahepatic ducts, then you have the extrahepatic ducts, and then you have the bile ducts itself. So first of all, we have the bile canaliculi. As I said, they do not have a lining of their own. They're formed by just the plasma membrane of the adjacent hepatocytes. And then you have the Herring's canals. They are lined by the simple cuboidal epithelium. After that, you have the interlobular bile duct and the interlobar bile ducts. These are lined by the simple columnar epithelium. And then when we talk about the extrahepatic bile ducts, these include the right and the left hepatic duct. As you can see here, the right and the left hepatic duct. Then you have the common hepatic ducts. Then you have the cystic duct, and then the yeah, that's it. And so the main feature of all of these, they are lined by the simple columnar epithelium, and between all of the epithelium, you have these structures called tight junctions. And they what do they do is that they prevent the leakage of the toxic bile into the blood. And the term which we call this is called the hematobiliary barrier. So, guys, do you have any questions about the bile production or the hepatocyte functions? No, so we'll move on to the last slide for today, which is gallbladder. So first of all, these are the two slides which are present in your slide box. As you can see, they have different shapes, so you cannot go on like, look at by your naked eye and identify the slides. These are different cuts. So when we talk about the differential diagnosis for these slides, first of all, the main one is fibromuscular layer. I'll explain what it is in the next slide. Simple columnar epithelium and irregular branched prominent mucosal folds. This picture, it's from your lecture itself. As you can see, it has the mucosal folds which are highly branched and anastomosing. So again, four layers same. You have tunica mucosa, and then the difference is the instead of tunica submucosa, you have tunica fibromuscular layer, and then tunica muscularis, and then you have the tunica adventitia. So when we look at the tunica mucosa, it is made up of very lightly stained simple columnar epithelium. As you can see here, these are tall cells which are really light in color. So this will help you easily identify the epithelium. Now, the most important one when we move on to the tunica fibromuscularis. So what this means is fibro and muscularis. So it is made up of smooth muscle as well as connective tissue. Here I have the zoomed in screenshot of the slide. As you can see, this part, this red part, it shows the smooth muscle. How do you know it's a smooth muscle? Because you can see the nuclei is elongated or stretched. So that's why you know this is smooth muscle. And the rest of this is the connective tissue. So I can show you this on the other slide as well. Here you can see, yeah, here. So you can see this is a smooth muscle as you can see the elongated nuclei, and then here you can see the loose connective tissue. Just a very important layer in diagnosing gallbladder. As soon as you see this, it's not present in anything else except gallbladder. Now, when we talk about the tunica serosa, it is made of the squamous mesothelium, loose connective tissue, vessels, and nerves. Now, this characteristics, which is the mucus glands, they might be present, they might not be present in our slide box. The slide, it's there. There sometimes in the tunica fibromuscularis layer, you can see the mucosal glands near the neck region only. Now, when we talk about the functions of the bile, functions of the gallbladder, I'm sorry, it is involved in the storage of the bile, increasing the concentration of the bile as well as the release of the bile. So, as I said before, it is lined by the simple columnar epithelium, which has absorptive function with the brush border as it absorbs the water from the bile to increase its concentration, and then it has a secretory function as I said, the characteristics are tall cells with light cytoplasm. So first function is that it is involved in the secretion of the mucus for protection. As I said, water absorption for bile concentration uh, increases its concentration, and then we have the tight junctions and the bile barrier. So tight junctions, as I said before, they're present at the upper part or the apical part, which prevents the leakage of the bile into the intercellular spaces, and this forms the hematobiliary barrier. And if it is disturbed by something, then this leads to jaundice, where the bile components enter the bloodstream. Now, simply, jaundice is basically a condition where your skin, mucous membranes, eyes, they turn up yellow because of buildup of the bilirubin inside the body. Yeah, I don't think so. There's anything else. So, do you guys have any questions? No, so that's it for today.
Thank you guys for coming. Let us know if you guys have any questions and see you next seminar.